experiment is going to demonstrate to us co-translation. So what is co-translation? Okay? So co-translation is the fact that the ribosome must be bound to the ER membrane in order for that protein that the ribosome is synthesizing at the time to get inside the ER. Okay? So the ribosome must be bound to the ER membrane in order for the protein to get into the ER. So this is how we do this co-translation experiment. So this is done in vitro, so in a test tube. In one test tube, you have all the components necessary for translation. You have your messenger RNA, you have your transfer RNAs, your amino acids, all that stuff. Okay? You throw it in, and ribosomes, of course, and you let translation go on. Now, as you can see here from the image, the ribosome translates the messenger RNA. We have our protein. These proteins have the N-terminal signal sequence. So remember, this N-terminal signal sequence is what you need. It's basically your ticket to get into the ER. Okay? So proteins with the N-terminus signal sequence have the ability to get into the ER, or at least they should. But now, so what we do is we let translation finish up. We have our proteins here in blue. They have their ticket, their N-terminal signal sequence. Remember, this is composed of about 6 to 12 mostly hydrophobic amino acids, represented here in pink. And now, to this test tube, you add microsomes. Remember, pieces of the ER. And what you see is that these proteins lack the ability to get in. They have the N-terminal signal sequence, but they cannot get into the microsome. Okay? So remember, in the first review session or the second review session, I mentioned this ability to get into the endoplasmic reticulum, the ER, is like trying to get into an R-rated movie. Okay? Children under 17 not admitted without chaperone or guardian, right? So these proteins are basically like you're under 17. They might have the ticket, but they still can't get in. So the ribosome in this case is going to act as the parent or guardian. You have to have your parent or guardian present at the time you enter your R-rated movie. So here, the ribosome must be present or attached, in this case, to the ER in order for the protein, which has the signal sequence, to get in. And that's exactly what we did then in the second test tube. So part 2 of the experiment is in another test tube. You add all your components for translation, messenger RNA, ribosomes, transfer RNAs, all that good stuff. But also at the beginning, within that step, you add the microsomes early in advance. And now what you see is that the ribosomes are able to bind onto the microsomes, and as they are translating the messenger RNA, the protein that is being synthesized is able to get into the ER. And there it is. So here's your mature protein now inside the ER because it had the chaperone with it, the ribosome. So the ribosome, this experiment proves co-translation. The fact that the ribosome must be bound to the endoplasmic reticulum membrane, so as it is synthesizing the protein can get in.